Hello, classmates of Nutrition 701, Protein Metabolism. Thank you for watching my assignment 4, in which I discuss a research article with you all. I will do my best. The research article I have chosen is Threonine Metabolism in Isolated Rat Hepatocytes, which are the liver cells of rats that have been removed from the rats. The particular researchers in the study or lead are House, Hall, and Brosnan. And I've left the reference below. And then, of course, it's in our Moodle online. So what's some background? What do you need to know before looking at the study to understand it? The main information we need to know and what will support our lecture are the 3 essential pathways of threonine. Threonine is an essential amino acid. It's indispensable, meaning we cannot use another amino acid to make threonine. We have to eat it. It breaks down, there's 2 major pathways and 1 minor pathway, so 3 altogether, all of which are happening in the liver, which is why hepatocytes are being utilized in this study. They either happen in the mitochondrial matrix or the cytosol of the liver, or a combination of both. The 3 pathways I have conveniently added on the slide below. There's a ketogenic pathway and a glucogenic pathway, because threonine can be both, and then another minor pathway. So the ketogenic pathway for threonine is going to happen in the mitochondrial matrix only. I think it's easiest to remember pathways if you're looking at the enzymes that they're using. So the first one is going to be threonine dehydrogenase. The threonine dehydrogenase pathway is going to turn into amino 3 ketobutyrate. Easy to remember because it's ketogenic. And the byproduct is glycine and acetyl-CoA. Acetyl-CoA, remember, enters that the Krebs cycle with a bunch of other ketogenic amino acids, and then part of the pathway shuts down because they no longer need to turn into acetyl-CoA. So that's going to be used in the place on that part of the cycle. So first major pathway utilizes threonine dehydrogenase. Second major pathway, it's going to be glucogenic, going to happen in the cytosol, and then lesser, but still in the mitochondria, so both. This is going to utilize threonine dehydratase, and you see or serine. So serine is going to use the same enzyme. So threonine dehydratase then becomes propenyl-CoA, and that is glucogenic, happening in the cytosol and then the mitochondria, still major. A minor pathway only happens in the cytosol. It's going to utilize threonine aldolase. And you're also going to produce glycine and acetaldehyde. Important to note, the minor pathway makes glycine. Also, the ketogenic major pathway uses glycine. The glucogenic pathway does not utilize glycine. That's important to know because that's going to be looked at in this study. So a good visual, this is the threonine pathway, and you see that it's happening in the cytosol and the mitochondria. The last one is going to happen, the minor pathway is going to happen just in the cytosol. And you see propionate, but propenyl acid, it's going to happen in cytosol and it's just going to happen in the mitochondria. The reaction is just going to happen in the mitochondria, make propenyl acid. And then acetyl-CoA utilizes first the cytosol and then the mitochondria. So both mitochondria, both cytosol, according to our lecture. So what other background information is key? Obviously, when you have a research question, first thing you do is see if other people have asked the same question. And the article gives alludes to 3 different studies. First of which is Bird and Nunn, looked at threonine in liver cells as well. And 87% of the metabolism from that threonine was happening via dehydrogenase pathway. So the first pathway in those normal fed rats. So normal fed rats, most of the metabolism is being done by that first pathway. 87 is a huge chunk. That doesn't leave much for glucogenic or the aldolase to really work. But in humans, by theory, that seems to differ. There's differing research in humans. So if we look at some of the information in humans, the Zhao, I'm going to guess, is the primary researcher et al. Adult humans didn't show any utilization of threonine to be used for glycine. So remember, pathway 1 uses glycine. So the ketogenic pathway, as well as the one using the enzyme aldolase, both use glycine. And it shows that adults weren't utilizing any of the specific isotope that they were using from threonine to convert to glycine. There was none shown, which 87% to none, does differ a lot. And then Darling et al. Showed low levels of glycine enrichment from threonine in low birth weight infants. So babies are showing very low levels of glycine that's been converted from threonine. So either that minor pathway or the major ketogenic pathway, which utilize glycine, there's very low levels of it showing. So in humans, we have none or low levels in infants versus rat 87% used from that major pathway. Mixed results. So there we go. So what are we questioning here? In theory, what I've highlighted, however, even low levels of glycine enrichment from threonine, when multiplied by the high level glycine disposal rate, can account for a significant portion of total threonine disposal. So by calculation, there should be more of it utilized in that glycine than what they were getting in humans. So is the threonine oxidation to glycine, is it higher than what was previously observed? So the aim of the study is, what effect does dietary protein levels on glucagon exposure have on the irrevocable loss of threonine? Where does it go? So essentially, if rat liver cells are fed threonine, then the rate of CO2 production or oxidation, that's what they're measuring with the CO2, the oxidation level, it'll be higher than previously observed in those studies, in theory. Both glycine dependent, so the ketogenic pathway and the aldolase pathway, and glycine independent, the glucogenic pathway, the oxidation will be observed by adding specific inhibitors. So there are inhibitors that are going to either inhibit the mitochondria or inhibit the glycine. So if the mitochondria is inhibited, you're mostly going to see the glucogenic pathway work, as well as your minor pathway at work. Or if the glycine is inhibited, you're mainly going to see the glucogenic pathway working only. So does that inhibit, are you able to see other pathways and the results of what threonine is still being produced or utilized or metabolized? So other things I looked at, the role of protein intake and glucagon on threonine oxidation. So what role does glucagon have? And the role of glucagon on threonine transport, specifically into intact rat hepatocytes. How long does it actually take to get there, be metabolized? So what did they actually do to test this? They have these questions. What's the oxidation levels look like with inhibitors, with different additives or substrates? So they took these liver cells specifically from male frog Solly rats. I've never worked with rats personally. My work typically deals with people directly. It's ethical. I'm not sure what the benefit of using a Sprague Solly rat is, and I don't know why males are used instead of female rats. If you can answer that in the comments from watching my video, it would be greatly appreciated on my end. I'm curious. So these rats, before their liver cells were extracted, were either fed a high protein diet with 60% casein or rodent chow, which is only 20% protein, not the good stuff. They either fed the good stuff or not so good stuff. So high protein or kind of low. So these cells were placed in a solution, suspended in a solution in flask, and they're incubated at 37 degrees, 20 minutes before the then added substrate. So the substrates included something called cysteamine, which would inhibit the glycine cleavage system, which inhibits the glycine. Alpha cyanosinamate, which inhibits the uptake into the mitochondria. Glycine, and then glucagon. So the incubations of the substrate were capped at 30 minutes, and then the CO2 levels were measured. The threonine oxidation was measured. Threonine transport was also measured. So following the addition of those substrates, the liver enzymes were or were not, so some were not, incubated with glucagon for 20 minutes. Followed by the addition of threonine. And then, so you have your cell, substrates were added, oxidation levels were measured, glucagon was or was not added, and then threonine was then added. And then those concentrations of that threonine to measure transport was measured at intervals of 5, 35, 65, 95, and 125 seconds. So after measuring at each of those different second intervals, the contents of the flask were transferred to centrifuge tubes that had like a silicone oil solution, and they were centrifuged for 15 seconds. And so when that centrifuge happened, the incubation media kind of was left on top in the oil, and the bottom layer was acid containing the threonine, so it separated. They measured for radioactivity using a method called liquid scintillation counting that I've never had to use before. And apparently the volume of the extracellular space carried down through the medium was used to find accurate threonine transport rates. So transport rates were kind of determined by using those second intervals to count, after each second interval, how much threonine was left based on how much they initially put in there. Why does this matter, right? What are the key results? And regardless of the results, what was actually significant? Significant shows if it matters. There was actually a significant increase in the oxidation of threonine and chow-fed rats specifically with the addition of glucagon. So only the chow-fed rats, ones with low protein, had increased oxidation when they had glucagon added to those cells. But also in the high protein diet rat cells, there was twice as high as a threonine oxidation rate anyways. So it was even higher. So glucagon made the chow-fed rat cells, which is not a lot of protein, makes sense, break down faster. Presence of sugar made it go faster versus the high protein rat diet. The ones who were fed casein had twice as high of a threonine oxidation rate to begin with. So high protein diet, there's more to break down. There's more threonine available. Duh. And then they wanted to measure also with glucagon. So glycine, so when they added glycine to the tube, with or without glucagon or that extra sugar, didn't matter. It wasn't a significant measure. So that did not matter. The addition of glycine made no difference significantly on oxidation levels. So to continue looking at some other key results, I take you to looking at the glucagon and the inhibitors. Some key things to note. So in the substrates that had oxidative inhibitors, glucagon still significantly increased oxidation when it was added. So in substrates that have oxidative inhibitors, glucagon still increased oxidation. So glucagon is still, when added, increasing oxidation. Alpha cyanosinamide specifically did not affect glycine oxidation, but did depress threonine oxidation. While the opposite was true with cysteamine. So it reduced glycine oxidation to less than the control, to less than the control level, but threonine oxidation was not impacted. So they work the reverse. So again, we see that across substrates tested, the presence of glucagon significantly affected oxidation rates. Now moving on to the discussion. The first observation. So the addition of threonine with added glycine did not lead to a significant reduction in CO2 production. Added glycine did nothing. And in theory, for those glycine-dependent pathways, the excess glycine should have been diluted and the total CO production reduced. The addition of cysteamine, the glycine cleavage inhibitor, did not significantly reduce the oxidation of threonine. So the estimation there is that the majority of CO2 produced during metabolism of threonine was not from those glycine-dependent pathways. So the oxidation was not reduced. So oxidation was coming from, even though glycine was inhibited, so it was coming from another pathway, a glucogenic pathway. The other observation that we see is the addition of alpha cyanosinamate, which limits mitochondrial use, ended up in reducing CO2 production to 29% of the control level. We interpret that 65% of threonine oxidation. Glycine-independent threonine dehydratase pathway is the primary pathway for threonine oxidation, which totally differs from the study saying that, what was it, 87%, 89%, or the high percentage from bird, and we can go back to that study here. Maybe. Totally differs from bird and nut, where they're saying 87% of threonine metabolism happens from dehydrogenase pathway in the normal fed rat. Versus if we're looking at just the liver cells, the explanation here is that, I lost my place, was that 65% of oxidation is actually coming from a pathway that does not utilize glycine. So your glucogenic pathway is actually doing the majority of the work, because we had an inhibitor that would stop glycine from, being utilized in the pathway. But we still got oxidation. So what is the conclusion there? We want to look at some of the strengths of the study. It's using specific inhibitors. So we can actually account for different types of oxidation pathways. Some things that I still want to know. Why did glucagon increase transport of threonine oxidation? It says it did, we know it did, but it wasn't really answered in the study. They theorized that it may possibly involve changes in the membrane. And other work needs to be done to look at that. There's, of course, as in every study we ever do, more studies need to be done, more research needs to be done. Future work is warranted to assess the impact of other amino acids, like those that have similar transport systems to threonine, and see if they affect threonine metabolism. So when you have more carriers available or less carriers available because our transport systems, because they're being used on other enzymes or other nutrients, it can decrease the absorption or delivery of that nutrient, right? The best example of this is if you delve in human nutrition, is calcium and iron interfere with each other because they both use the same uptake transport system. And as a result, if you're drinking milk with your iron pill, you don't absorb all of that iron because some of it's using for calcium transport. Similar at this, what other amino acids are going to use similar transport systems? And if they're being metabolized at the same time, does that decrease threonine metabolism? And the final takeaway for me from this was glycine-independent pathways, or dehydratase, or your glucogenic pathway, is dominant for oxidation of threonine, 65%. And then oxidation is increased by protein-fed diet and glucagon in liver cells. So I'm going to go over that again. Not the ketogenic pathway, but the glucogenic pathway using your enzyme dehydratase was found to be the dominant pathway, shockingly, 65%. And then things that are going to decrease increase threonine metabolism, the high protein diet, at least in rats, and the addition of glucagon in those liver cells will help that threonine be metabolized. That concludes my presentation. If you know why male rats are being used instead of females for their cells for studies, please let me know. I'm very interested. Bye for now.